Hello! So this lesson is going to be a little bit different because this is a practical lesson. The topic of this lesson is on how to move bacteria around, microorganisms around safely in a laboratory. Now in an ideal world you would actually be doing this in a laboratory, but that is not possible to do through this video medium. So what I'm going to do today, and I've never tried this before, is to do this practical at home, try and improvise all of the equipment and see if I can show you the techniques from my kitchen. Okay, that's the challenge for today. What you need to take away from this are the steps in safely moving microorganisms from one space to another. You need to know what equipment's being used, you need to know why it's being used, you need to know how it's being used, and you need to know the 3 steps: pre-inoculation, inoculation, and post-inoculation. Enjoy. Okay, so the first thing you normally need to grow bacteria in a lab is some sort of nutrient for them, either in the form of a liquid broth or a gel plate. Now, of course, we're not in a lab at the moment, so I don't have those things, but I think I might be able to make them. So fortunately, I've got a little bit of leftover chicken stock from making some stock previously. Now that contains a lot of the important nutrients that bacteria will need. Just like any other living organism, they're going to need a source of carbohydrate, they're going to need a source of protein, they're going to need a source of fat and essential vitamins and minerals. Now chicken stock will have most of the minerals, vitamins and protein and fat they need. It might be a little bit lacking in the carbohydrate they need for respiration. Now of course, I don't know what's in here, I don't know exactly the chemical composition of this, so I'm kind of making this up as I go along. But one thing that's really good for bacterial growth is simple sugar. Okay, and this is a simple sugar. This is maltose. This is for home brewing. It's basically a different form of sugar from the one you get in your bags of sugar. You could use that, I guess, but this one, maltose, is literally pairs of glucose molecules stuck together, and it's the simplest sugar I have. So if bacteria are going to grow on anything, and they're not really that fussy, it's probably going to be this stuff. So I'm going to take about a tablespoon, again, completely guessing here. I'm going to add that into my chicken stock. I've got about 200 millilitres in there of the chicken stock, and I think, just looking at it, that's probably a bit concentrated, a bit gloopy. The other thing I'm worried about is if it's too salty and too strong, it's going to kill the bacteria by osmosis. So I'm probably going to add about the same amount of liquid again to make that up to 400 ml or just under half a litre, so that then I can heat that up and have something a little bit thinner that's going to go into the next step. Okay. Okay, are we hot enough yet in here in the microwave? Let's get the old thermometer in there, see what it says. No, that's not getting up to boiling. No, not convinced. Right, the broth with all its nutrients is in the microwave heating up. I'm going to boil that to make sure it's sterile. Now, this little jar here has just been through the dishwasher, so it should be sterile. And when I put the boiling nutrient broth in there, that should keep it nice and sterile as well. So if I keep the lid on and keep that nice and closed, that will be a sterile liquid nutrient broth ready for me to inoculate or add any sort of bacteria culture or yeast culture that I wanted to add in there. So that's the liquid nutrient done. I'll do that when the broth comes out. The other one is slightly more common, is the so-called agar plate. Now agar jelly, this stuff, which I luckily have thanks to a Christmas present from several years ago of one of those molecular gastronomy kits. Luckily I've got a few sachets of the agar jelly here. This stuff is essentially derived from seaweed, and all it is a gelling agent. It's like gelatin or any other gelling agent. You mix it with something, and as it cools it will set like a jelly. Now normally that would be the proper nutrients that you would have in the lab. Normally this would be done for us if we were doing this in a science lesson, but as we're all at home, this is what we've got to do to make sure this works. This is a bit of an improvised version. So I've got the agar, the nutrient broth is heating up. Just like any other jelly, what I would do is once I've got rid of the bit for the broth, I would add the gelling agent, make sure that's nice and hot. I would normally pour it into Petri dishes, but I don't have Petri dishes. What I've got instead is I've got these lovely food containers, right, which have just been through the dishwasher, so they should be relatively sterile as well. They've also got nice lids that I can put on the top of them. Okay, so those are going to be my Petri dishes. I've got the agar, I'm going to make up the nutrient broth and I'm going to pour those in there and see if it works. But as I say, this is completely experimental. I've never tried to do this at home. There's 2 grams of agar in here. I reckon that should probably gel about 125, 150 millilitres of liquid, but we shall see. Well, it's not boiling, but it is still going up. All right, so stabilised at 93. But I think considering this is going in a pan for the agar, that is warm enough to consider sterile for now. Okay, nice and hot then. So I'm just going to pour off a little bit into my broth jar to be my liquid nutrient broth. There's that. Okay, so that's my liquid broth. Hard to do and film at the same time. So there's my liquid broth. That's going to go to one side. That is all the nutrients that bacteria need. So before this cools down too much, I'm going to pop this straight in to the big pan, trying to get too much on the hob. There we go, I've got my heat probe. I'm going to get that heat on, nice and high. Get that heating up. Feels like a really weird Jamie Oliver program. Okay, that's heating up now. Here's my little sachet of agar. It's quite old, but I don't think it'll mind. Now that's boiling away. So, I'll turn that down a bit now. Sort of boiling over. Open this up. Okay, so you're bubbling away nicely. So this is going to go straight in. In we go. Got to stir that until that's all nicely dissolved. Now, of course, you're getting an insight here into what normally goes on in the prep rooms to make your lessons work. So next time you see them, do thank your technicians. I'm just going to keep stirring that until it's completely dissolved. And this is where when I pour them in, they will melt really embarrassingly and something stupid happens like that. It's really interesting doing this actually because agar has a really specific smell, at least agar plates, agar jelly has a really specific smell, and doing this I'm now getting an impression of which component smells of which, and this smells pretty much like agar jelly, which is not a massively scientific inducement for this, but so far so good I reckon. I'm going to try and do this and keep the close-up camera rolling. So, here we go. Roughly half, or roughly a layer in there, and a layer in there. I'll keep that bubbling away. Hopefully that will set and start to gel. Okay, so we're going to leave that there. Hopefully it will set, hopefully it will gel. Have a look there. I'm hoping I've boiled it long enough and that those are bits of chicken stock, not bits of agar. Only time will tell. Okay, I made a little executive decision here. Looking at the volume of the liquid, is to add another gram of agar. Again, we are really improvising and experimenting here, so I have no idea whether that's going to be the right one or not. But I've also found another spare Petri dish. Spare improvised Petri dish. So, right, got some lovely agar gel coming on here. That little lump that hasn't dissolved in the saucepan, that's making me a little bit nervous, because if that's agar, then maybe this won't set. Right, that's my jelly poured. They're in the moulds there. So you can see I found a third one to use as well. So a little bit too much liquid. So I add that extra gram of agar. No idea if it's going to work or not. Never tried this before. But they're all poured. At the moment they're sterile, of course, because they're really, really hot. As they cool, of course, they could become contaminated from the air, from all sorts of different sources where the bacteria might come from. So I'm going to put some lids on, try and keep them sterile, get them in the fridge and see if they can set. Right, here's hoping. Now, next thing you're going to need to put your bacteria from where you want them onto your agar is a thing called an inoculating loop. Now, of course, I don't have an inoculating loop because they're all in my lab and that's not where I am. So I'm going to have to try and improvise a sterilisable metal loop that I can heat up and cool down to burn any bacteria off it, and then use it to move microbes around the place. So that's the next challenge then, find myself an improvised nichrome loop. Okay, I think I have a solution. What I needed was a wire that I can heat up and cool down quickly so it becomes fully sterile, right, so I can move bacteria around with it but doesn't melt. So I was trying to think what I've got that might be non-meltable but easily able to heat up and cool down. And then I remembered in the back of the shed, sparklers, right? Sparkler wire is specifically designed for that purpose. Now these are sparklers that have still got the burny bit on them, so I'm going to have to get rid of that before I can make my inoculating loop. Oh well. This is entirely for science. Well, that was fun. Right, as it turns out, I can probably get away with just the handle part, so that was entirely unnecessary, but, you know, who's counting? So, what I'm going to do, I'm going to try and make inoculating loops out of these two bits of wire here, as you might be able to see on the close-up cam. I'm going to do that by taking the little bit at the end, the handle bit, I'm going to cut that off, and then I'm going to try and wrap that around the end of this screwdriver here to make a little loop that I can then use to pick up bacterial colonies and move them to one place. Now this material should be absolutely perfect for that because it can get really hot, so sterile, but I've just shown you it's not going to melt when it's on fire. Okay? Right, so let's have a go at that then. Right, so just cut the useful bits of wire off. Like so. And then I wonder how flexible this stuff actually is. Pretty flexible. Well, not the best inoculating loop I've ever seen, but it will do the job for our purposes. Now, obviously, because I don't want to get burnt, I'm going to make a improvised insulating handle. If we get results out of this, it's going to be absolutely spectacular. So I'm going to wind that up. Right, that pretty much is a good facsimile of an inoculating loop. So we've got our gel and our broth, and we've got our inoculating loop. Now I just need something disgusting to test. Well, this looks pretty disgusting. Maybe I could start trying to hoik some of these nasty things out of there. So this is a pot of pepper dip, which obviously got forgotten about in the back of the fridge. I was going to throw this away, but from a microbiologist's perspective, we could see what's growing in there. How disgusting am I feeling? Okay, so far so good. So now we can go on to the actual making up and testing our bacteria plates. So first stage, and the one you should write in an exam answer, is called pre-inoculation. Now in pre-inoculation, we do all the stuff we've already done, and we sterilise everything around the area, so we're not getting anything other than the microbes that we want. So this is pre-inoculation. Right, so the equipment you need for the actual bacterial transfer, if we were in a lab, you need to do all work by a Bunsen burner. Now that basically is so that no bacteria falls in from the air, because you've got a convection current moving all the bacteria away, but it's also so that you've got something to sterilise necks of bottles and your inoculating loop in. I don't have a Bunsen burner, of course, so I'm going to be using this blowtorch, right, which I happen to have for reasons I shall not explain. I am also going to be using my inoculating loop, as made earlier, which I couldn't be happy with that. That's brilliant. And also, we have agar plates. These have gone absolutely brilliantly. Okay. If I grab my close-up camera and show you, they're solid. They're nice and solid. That looks like agar jelly. It's not flopping around at all. Okay, and that's a perfect surface, it looks like, to grow bacteria on. However, whether it will work or not as agar jelly, I have no idea. So, we got our agar, we got our inoculating loop, we got our blowtorch. What are we going to try and grow? Well, I have this disgusting, out-of-date, roasted red pepper dip, which has gone all manky. We're going to see if we can identify some of the microbes on there. I've also been, as many people have been, trying to start a sourdough starter. So this is a sourdough starter here. Now sourdough is usually just a combination of yeast, water and flour. I've been trying to do this with very low amounts of flour because that's been hard to come by for the time being. So whether or not I've actually got sourdough in there, a sourdough starter in there, or whether actually I've put so little flour in there, really all I've got is fermentation, don't know. But there's certainly not a microbiology going on in there, and I might be able to find something. Okay, I'm going to test some soil from outside. I'm going to do some fingerprints and see what I can find. Okay, let's do it. With pre-inoculation done, all my kit ready, desk scrubbed down, hands washed, got my Petri dish and my agar jelly in there. And what I'm going to do is I'm going to write on this what I intend to put on it. Now you should be able to just turn these upside down because the jelly should hold that nicely in the dish. I'm still a little nervous as to whether these are going to work the same. We always write on the bottom of our Petri dishes, reason being lids can move, lids can get lost, lids can get turned around. The bottom should tell us what it is. So I'm going to draw a line straight down here. I grab the close-up camera so you can see what I'm doing. So I'm going to draw a line down the middle, and I'm going to maybe divide this into 6 portions like that, and then I write on what I'm going to do. So this one here is going to be my dip, this one here is going to be my sour dough, this one here will be a fingerprint, this one here I reckon will be writing over the lettering there, that one there will be the soil. As I say, we do all our work by a Bunsen burner. That way we keep everything nice and sterile. And when I say by a Bunsen burner, I don't mean like this, I mean like this. Okay, this is how close they need to be. I'm going to start with the dip. Hopefully we'll be able to see what's going on if I just put this camera on, like so. See everything there. Okay, so the trick is to keep the lids on as long as we possibly can. Now, obviously in a lab, you'd be maybe in an apron, maybe with gloves, so on and so forth. We don't have that. So let's run our Bunsen. There we go. Now I can pretty confidently take the lid off my agar plate and just put that to one side. Now if there's a Petri dish, I'll just lift it and put it back, but it isn't. Next step, now this is our inoculation step, I'm going to sterilise my inoculating loop. So I'm going to pop that in the flame. The hottest point of the flame being just at the tip of the blue circle. Now if this was a nichrome loop, you'd do this until it went red hot. This one I'm not going to do that much because that would be perfectly adequate to sterilise it. And I'm not going to go straight onto my bacteria with that because of course that's red hot, so I'm going to kill the bacteria if I do that. So I take a corner of my agar jelly and I touch it down and I get a little noise. That's the most fun bit of this experiment. I get a little, so that's now sterile because I know that's sterile. I know the fire would have killed off the bacteria. So now I take my lid off my sample. This is pretty disgusting, so we'll see what happens. And I'm going to hoik out what looks like a penicillium fungi. Now I don't know if it is, but I'm going to just sort of see if I can get a bit of that on there, and then I go onto my jelly, and I don't dig, I just wipe, just gently, like that, just gently, like that, onto my area that I'm interested in, like so. Put the lid back on to stop contamination, make sure the lid's on there, and then I sterilise my loop. And that's the inoculation step. Okay? I'll do that one more time with the sourdough. So we've done our pre-inoculation, everything's nice and sterile. So we go, heat the loop, like so, and that's red hot now. Now we cool the loop, and you might be able to hear the tss. Oh, that was a good one. Now as a thicker loop, it's taking more to sterilise, to cool it down, which is a bit unfortunate. I'm going to take the lid off this, and then, where did I say sourdough was? Over there. I'm going to take a little bit, actually off the lid I think, of this one. I'm going to take a little bit off, the lid like that, and I'm going to just wipe it onto the agar jelly, like so. Okay, lid back on, sterilise our loop. Now as you can see, I have got that red hot, you can see down there. I'll cool that off on the agar before I go. Right, I'll come back to that in a moment, but the last steps, the finishing off steps, are post-inoculation. What we would do is we put the lid on. Now these tubs are going to go properly sealed, which is not what we want, because obviously bacteria are breathing, living organisms. Now on a Petri dish, the lid would fit nice and loosely over the top, so let air, let oxygen in, carbon dioxide out. With this one, I'm going to have to improvise again, and we would usually take them to an incubator, and we would store them upside down like that. Reason being, they are going to give off moisture. That moisture will hit the lid, and it will fall down if they're the right way up. We don't want that, that will smear our bacteria around. So we store them upside down in an incubator. Now in a school lab, we're only allowed to do those up to about 21 degrees, 21, 25 degrees, because we don't want the speed of growth that would be achieved at say 30, 35 degrees. But in industry, in industrial conditions where they're a bit more controlled, they can go a bit warmer, a lot hotter than that, and they can get much more rapid growth. Now obviously I don't have an incubator, the closest I've got is my conservatory. So I'm going to finish off taking a few samples on a few different ones of these, and then I'm going to pop them upside down in my conservatory, and we'll see what we get. Right, now to test my fingerprint, I'm just going to lay my finger very gently on the jelly. And that'll do it. Put it on. Okay, I've pre-sterilised this, so let's see what the chili soil has waiting for us. Once and on. Lid off. Gentle bit of smearing. Dabbing off there. Now that bit's obviously got a little bit of contamination on it as well, just shows how easy done it is. And then just sterilise our loop again. Okay, so that's how you would safely move bacteria around from one pot to another pot. There is one last little bit I have to show you, which is what we would do with a broth. If we were going broth to plate, plate to plate, sample to plate, doesn't really matter. The only difference with our broth is if we were going to take something hygienically out of here, we would have our Bunsen burner running, like this. We'd take our lid off and we would flame the neck, like that. Okay? And what that does is it prevents any bacteria that have got, or contamination that's got onto the rim of the neck, the lid, to get into the bottle. Okay, so that's just the only difference with broth. So for your purposes, we've got pre-inoculation, sterilising your desks, making sure you've got sterile agar and a sterile inoculating loop, and doing all your work near a Bunsen burner. Inoculation, Bunsen burner on, heat your loop, cool your loop, scoop your sample, spread it on your patch, keep the lid on nice and tight, and then post-inoculation. You get the lid on with a single piece of tape, you don't seal it up because they need oxygen, right? And then you turn it upside down and incubate at 20 to 25 degrees, somewhere nice and warm, for maybe 24, 48 hours and see what you've got. Okay, and that's your 3 stages: pre-inoculation, inoculation, post-inoculation, and that will be your practical for this week, and that is today's lesson. I'm absolutely delighted at how that's gone. I think that's gone really well. I will add on after this how the results have come out. Thanks very much.